Welcome back to cell communication. Before we get started, let's go over what we already know. Cell-to-cell communication is essential for multicellular organisms to survive and function properly. There is 3 types of signaling: direct, local, and long distance. We've also gone over the signal transduction pathway, and it consists of reception, transduction, and response in order to trigger a specific cellular response within the cell. Let's start out with the signal molecule that starts it all. So what is this signal molecule? Well, technically, we call these ligands. And these ligands have a lot of roles in the importance of the cellular interactions. So for instance, let's take this graphic I've drawn here. This, where I'm highlighting in yellow, is the plasma membrane, and the red U shape is a receptor, while the green little dot is a ligand. A ligand travels at random, so we will do some dots right here to signify a ligand traveling at random. But once the concentration is high enough, meaning that up here is high enough to where it can bond to this receptor protein, a ligand will bond to this protein, allowing for the reaction to start. So here are a few things that should be known about ligands. Ligands activate a protein for a short amount of time and are then recycled, which allows for biological control of many interactions. Also, the amount of time a ligand spends attached to its receptor or specific protein is a function of the affinity between the ligand and the protein. A high affinity indicates the ligand will tend to stick to the protein and modify its function for longer. So high means longer, and vice versa. A low affinity for the protein will be less likely to bond in the first place and will release from the receptor faster. So not as long, if any, if the affinity is lower. Now, when a ligand bonds to a receptor protein, a shape change occurs known as a conformational change within the receptor protein to have a better fit for the ligand. This is also known as phosphorylation. Now we see, so I don't mess up the spelling. Which regulates protein function and cell signaling by the changes of the phosphorylated protein. Now let's talk about pathways. First, we're going to start out with the G protein pathway, which is the most common pathway used by cells. This pathway consists of a G protein linked receptor and a G protein. The G protein linked receptor or G protein coupled receptor is found on the membrane of the cell. It consists of a large family of cell surface receptor proteins that share a common structure and method of signaling, which is only found in eukaryotes. One of the most important characteristics of a G protein coupled receptor is it has 7 transmembrane alpha helices, as shown right here. Some information to note about the GPCR is it is the attachment point for the ligand in this pathway. Like previously stated, the shape will change depending on ligand attachment. No ligand means the GPCR is inactive. These help regulate growth, immune system, our sense of smell, taste, visual, and behavioral mood. Now let's talk about the G protein. All G proteins that associate with GPCRs are heterotrimeric, meaning that they have 3 different subunits or functions. These 3 subunits are the alpha, beta, and gamma. The alpha and gamma subunits are attached to the cell membrane through lipid anchors. In this graphic, the alpha is in red, the beta is in blue, and the gamma is in yellow. G proteins come in different types, but they all bind the nucleotide guanosine triphosphate, or GTP, which breaks down to form GDP. When a G protein is attached to GTP, it means it's active or on, while a G protein that's bound to GDP is inactive or off. Now let's do some practice with a ligand present. When a ligand attaches and phosphorylates the protein, the G protein alpha subunit exchanges GDP for GTP. The alpha subunit dissociates from the beta and gamma subunits and triggers a cellular response. GTP is hydrolyzed to GDP and the signaling molecule comes off the receptor. The GDP also creates a phosphate when it is converted, and the ligand is removed. The alpha subunit comes back together with the receptor and the beta and gamma subunits. And as you can see, this whole process is back to its original state or recycled. Another important receptor protein pathway is the tyrosine kinase pathway. These play an important role in a variety of cellular processes, including growth, motility, differentiation, and metabolism. A kinase is just a name for an enzyme that transfers phosphate groups to a protein or other target. A receptor tyrosine kinase transfers phosphate groups specifically to the amino acid tyrosine. One special ability about this pathway is it has the ability to act as a catalyst in order to rapidly relay proteins, 6 at a time. These are essential for the body but must be regulated because overactive growth has been linked to some cancers. So how does it work? First, bind the extracellular domains of the two nearby receptor tyrosine kinases. The two neighboring receptors then come together or dimerize. The receptors then attach phosphates to tyrosines in each other's intracellular domain. It uses ATP for phosphorylation. ATP is made up of 3 phosphates. The kinase takes one of the phosphates to phosphorylize. Eventually, all phosphates will be phosphorylated. These Y-shaped things act as the tyrosine residues. The paired receptors phosphorylate each other on tyrosines in the intracellular domain or the region inside the cell. These phosphorylated tyrosine residues are found in green. This triggers a cellular response. Next, we will be talking about ion channel receptors or ligand-gated ion channels. These membrane receptors can act as a gate that open or close when a specific ligand binds to it. These allow specific ions into the cell, and they are only activated by a ligand. The middle of the channel is hydrophilic, which allows for ions to cross the membrane without coming in contact with the phospholipid's hydrophobic core. These are very important in the nervous system and our brains. Let's look at this depiction on the membrane. With the signal molecule present over here, ions are allowed to flow through this open channel effectively due to the signal molecule. On this side, however, the channel is closed. Let's do a little better depiction. I don't know if you can even see that. Because there is no indication of a signaling molecule on the signaling molecule binding site, which means no ions can pass through. And this is the basics of an ion channel receptor, or ligand-gated ion channels. The last pathway we will be discussing is the intracellular receptors. Unlike the other pathways, these receptor proteins are found in the cytoplasm or nucleus of the cell. These differ from the others because they are lipids, meaning they don't need to be on the plasma membrane. In order to pass through the cell membrane, the ligands that bind to these receptors are small hydrophobic molecules. So this black line is going to act as the nucleus line that separates the nucleus from the cytoplasm and the rest of the cell. And these red dots are going to be the hydrophobic molecules that enter into the intracellular pathway. So let's note that it happens in the cytoplasm of the cell. Okay, so I redrew the graphic to make it more focused, but as you can see, this hydrophobic molecule is going to travel through the plasma membrane, and it's going to meet the intracellular receptor in the cytoplasm of the cell, and then it's also going to travel into the nucleus to land on the DNA. Now, remember, these receptors are mostly for receiving hormones and steroids. So this example is a hormone entering the cell. The receptor shape changes due to the binding. So I'll make it more known, but the receptor changes its shape. I'm going to do a better graphic. Due to the binding. And this creates a hormone receptor complex. So a hormone receptor complex, which permits the translocation of the nucleus and bonds to the specific part of DNA, which is right here. Now, this allows for the regulation of gene activity, and these hormone bonds allow for certain regions of the receptor to be exposed, allowing for the attachment of specific sequences of DNA. Now, this is important because it alters the level of transcription. And becomes a transcription factor. Now, this is the copying of DNA of a sequence into an RNA molecule. The RNA molecule is then modified in the nucleus to make a mature mRNA, which is used by the cell to make similar cellular functions. Our last section will be on secondary messengers. These messengers are relay molecules located in and throughout the cytoplasm of the cell. And they greatly serve to amplify, so to speak, the strength of signals. One of the most common types is cyclic AMP. Adrenaline is a common hormone that achieves its effects through cAMP. Cyclic AMP is synthesized from ATP by the action of the enzyme endinyl cyclase. The rise of cAMP turns on the appropriate response in the cell by either or both, by changing the molecular activities in the cytosol or turning on a new pattern of gene transcription. And here is a graphic of what cyclic AMP looks like. Next, we'll talk about calmodulin and IP3. IP3 is a soluble molecule which diffuses throughout the cytosol and binds to receptors on the endoplasmic reticulum, which causes the release of calcium ions into the cytosol. This rises in intracellular calcium triggers the response. Likewise, calmodulin is found in the cytosol of all eukaryotic cells and interacts with many proteins in the cell. It is involved in a wide variety of processes including cell regulation, intracellular signaling, phosphorylation, fertilization, but most commonly muscle contraction. Calmodulin can bind up to 4 calcium ions. One of its functions includes activating calcium pumps. These pumps remove calcium from the cytoplasm by either pumping it out of the cell or storing it in the endoplasmic reticulum. This control of calcium in the cell produces the cell's response. Thank you for listening, and I hope you learned a little bit more about cell communication.